So, okay, that's good. So, like Farzan mentioned, one of sort of my broad interests is in trying to understand the organization of the cell. And as we keep learning more about this in recent years, we are finding out that the cell is remarkably spatio-temporally well organized. And I will show you some motivation on how the cell's organization has sort of spurred projects that are closer to both soft matter abstract, but also more focused problems based on particular biological phenomena.
It's on. Okay, good. So if somebody asks a question,
thanks. Okay. So that's our first NAFU for the day. That resolved quickly. So that's good. So the motivation for a lot of the questions I'm interested in is in thinking about how the cell is organized. And the eukaryotic cell, caricature of which is found here, is this remarkably sort of well-organized system. And one mechanism by which this compartmentation happens, which in turn promotes specific processes to happen at the right point of space and time, is through membranes. And these are ones we're familiar with. The mitochondria is a membrane-bound organelle. The cell itself has an outer cell membrane that helps separate the exteriors from the interiors. And this has been long known to be a mechanism of compartmentation. What's been known for a couple of 100 years is also that the cells contain many compartments that are enriched in very specific combinations of molecules. Examples of which are given here. These are dozens of different compartments. And all of these compartments lack membranes. So for a long time, how they form and what keeps them organized has not been well understood. But clues onto how they form have been sort of piling up slowly over the past century. So here are some lovely old papers that I thought I'd cite. One is from Montgomery, who wrote this 400-page manuscript on the nucleolar structure. And he seemed to notice that the nucleolus not only lacks a membrane, but it seems to behave like a distinct material with a separate refractive index, that it seems to dissolve and then reform across the cell cycle. E.B. Wilson, sort of considered the father of modern cell biology, wrote a speculative article in 1899, arguing that the cytoplasm was nothing more than a mixture of liquids of different identities, much like a.
So the nucleolus, remind me, is believed to be concentrated messenger RNA and ribosome?
Yes. So the question is, what exactly does the nucleolus is made up of? So the nucleolus is one of these compartments that's formed around ribosomal DNA, and its function is thought to promote transcription of ribosomal RNA along with the assembly of the ribosomal unit itself, which involves both the RNA that's produced actively as well as proteins that are imported from the outside. So one of the things we've started to understand really in the last few years, and this is for a lot of people's work, but mostly led by Cliff Brandsman at Princeton, Tony Hyman and Mike Wilson, is that these compartments are now thought to be sort of more or less forming by the process of phase separation. And to this audience, I don't need to explain what that means, but I thought I'd show you a video, which is kind of captivating. So here, the outline is mild, but you will see a cell's outline here. And as the video plays, the cell is undergoing arsenite stress. And there are two proteins that are tagged in green and pink color. You will see what happens when stress forms. You can see here that these proteins form or demix, condense out of the cytoplasm into these droplets that dance and coarsen. And there are two different kinds, at least here, the green and the pink ones. Their names are not particularly important. I'll play that again, and you can see that the behavior of these droplets are very reminiscent of what you might classically think of as liquid-like droplets. And in fact, this paradigm has grown to be important in and sort of fairly universal in trying to explain how these compartments form, that they're like liquid droplets in the cytoplasm. Sorry, this is just a mirror. So this is a mirror image, I think. Oh, it's here. Yeah, I think so. Let's just focus on this cell. I'm not completely confident. Yes. Thank you, David. Yeah, so what's become very popular in the last few years is to invoke this language that's based on oil and water phase behavior to explain what happens here. And this is a very zeroth order and not a very universal approximation for the following reasons. The cell doesn't consist of two liquid phases that coexist, right? There's really dozens of compartments that coexist. And here is an example of a nucleus where three different components are stained. And you see that in green, in yellow, and in red, and my apologies to the red-green colorblind, there are distinct compartments here that form and coexist. So the cell is really this fluid compartment that has multiple phases that coexist with each other, with at least two having distinct composition.
What is arsenide stress? Is it something arsenide powder?
The question is, what is arsenide stress? Yeah, so you add sodium arsenide into the mixture, and that poisons the cell, and then it upregulates a stress response, parts of which involve forming these pink and green bodies, which are called stress granules and T-bodies. Yeah, and so none of these are stress. These are endogenous. They're found during the cell's healthy physiology. We don't want to go into the names, but there are multiple of these condensates that coexist. So there's a lot of interesting questions we might ask from a soft matter and physical perspective on how distinct compartments can coexist and what are the features that let this happen. I do not know what the question is it similar to an SOS response in bacteria? I don't know the answer. I can tell you that mammalian cells have many, many different stressors. And in response to wide varieties of them, osmotic, salt, crowding, the arsenide stress, etc., all of them give response to certain compartments they form. Whether these are identical is yet to be sort of figured out. Yep. Another thing I want to point out, though I really won't go into this too much more, is that these droplets do not have to be like classical, oil, water, liquids. They can exhibit complex morphologies. Here is a picture of a nucleoli isolated from the oocyte of a frog. You can see that it has this nested sort of Russian doll-like structure where the green droplets are embedded in the red droplets. And they have other complex interstitial properties. And in general, these are fairly widely spanning materials. They don't have to be liquids. They could be gels, glasses, solids. They often form or near surfaces like membranes or chromatin or RNA. And they're also often sites of activity, whether chemical or mechanical. So I guess the slide is to motivate that there's lots to learn about the biology, but also as a physicist approaching this problem, these are questions of multi-component fluids, which are often active and exhibit multiple phases. So there's lots of things to learn. In this talk, I'll focus on two specific questions. One is, what can we learn about the phase behavior in fluid mixtures that have very many interacting components? And a model that I think I will introduce that's been kind of fruitful in thinking about this is on mixtures that have components with random interaction. And then the second one is a problem that's much more focused on a very specific biological system, and that's how does active processes affect multi-component phase separation. And I'll focus particularly on how RNA activity influences phase behavior. Okay, good. I'll start with the first part, and I really don't need to get through both. So if there are questions, we can stop. But the first part is a piece of work that I've been doing with Michael Brenner here at Harvard. And there's a preprint if you want to look at it. So the question again is, if you have a mixture, fluid mixture, with very many interacting components, is there something we can predict or say about the general principles that govern its phase behavior? And this has been a question of much interest for many years now, but it's been hard to assay systematically for systems with more than a handful of components. And the reason why it's very hard, there are multiple reasons, but one of them is it's just numerically hard to explore this because there are multiple constraints that are set by thermodynamic equilibrium, which are nonlinear. And it's not been easy to investigate this systematically for large systems. Another reason why it's hard, as you might imagine, as you explore the number of species, the design space grows up. And so it becomes hard to survey the system. And then even though this is steady state, there are also multiple dynamical regimes which become hard. So there's a number of reasons. Yeah, the Gibbs, the question is, can you not use information about the Gibbs phase rule? Yeah, so the Gibbs phase rule is a constraint it's not, even though it has the name rule in it, it's not an actual rule. All it says is that thermodynamically at equilibrium, if you have a system with n components at fixed pressure and temperature, you can't have more than n cases. It's not prescriptive of what a system will actually do.
The observations that you made with more than 5 components of this kind
Yeah, so the question again is, if you have more than 5 or 6 components, is it consistent with the phase rule? What I can tell you is that, at least from what we have learned, they're definitely, they obey the constraints of Gibbs phase rule, but they might not even reach the theoretical prescription of n cases under the conditions we study. In general, it turns out to be hard to explore. And of course, the Gibbs phase rule is only true at equilibrium in infinite systems. So when you have finite systems and you have activity, these things may not work. But it's a good question. Thank you, Sam. Okay, so because this is kind of hard, we decided that we're going to reformulate this into an accessible system with the constraint that it's still somewhat general, so we can learn something from it. And so the reformulation we do is the following. We still have multiple components, but instead of allowing them to be arbitrary, we say that we start with equal concentrations of all of them, and that we sample their interactions from some random distribution. So this might be, your favorite library of molecules that you design. It could be a bunch of colloids that you have in your cabinet, and you just go randomly sample a few of them. The question now is, does making this approximation help you make any headway along this problem? And the reason we do this is actually based on a lovely little note that Sears and Quester wrote about 20 years ago, which initially explored whether random matrix theory can help us think about it. And so it remains fairly general because there are still interactions, there are still multiple components, and with tricks we can employ that I'm not going to talk in detail, it makes us accessible to solve these nonlinear coupled phase field simulations. Like I said, I've reduced the design space because I've imposed equimolarity and I've imposed interaction from a distribution. And the final assumption is we're going to focus on Spinodal decomposition. So a lot of these, some of these approximations beg the question, how does it translate to the cell, for example, where nucleation might be more important. That's not something I'll address today, but making these approximations allows us to start to make progress towards this question, and we can still learn something. That's the hypothesis, the claim I will make.
I agree with all that. Presumably cells have had 2 billion years of evolution to fine tune their parameters.
Absolutely.
So null model.
Right. So the question again is that cells not only may have been different regime, their interactions have probably been fine-tuned over multiple billion years. And indeed, that's true. And so this is just a starting model. And there's a variant of this I've started to explore where you can impose design constraints and then ask what happens when there's design and crosstalk. But today I'll talk about a null model, if you will, of what random interactions do. And it's still not a completely sort of basic null model, because you could still have a library of molecules whose interactions are described well by some distribution. And there are correlations. That's the only complication. So if you assume they're uncorrelated, this still turns out to be a good model. All right. And so the hope is by doing all of this, can we start answering these questions on how many cases might exist? Can we make any comments on the composition? Because it's a dynamical simulation, can we learn something about dynamics? And then can we make corrections and start asking what happens with activity? Okay, so I'll just quickly walk you through what the model looks like. We start by picking the number of species n, and then we sample their interactions from a normal distribution with some width. This chi-ij parameter, it's not strictly the interaction between two molecules. It's proportional to something like the virial coefficient, and formally it's an exchange interaction. And we can write down a free energy for the system. This is sort of the most simple free energy that allows for interactions between species on top of entropy. With this, we can simulate dynamics by just stating that we follow the evolution of all the concentrations subject to diffusive fluxes in the chemical potential, or model B dynamics. And this also enforces overall conservation of mass. So we're studying the closed system and how it evolves.
So equimolar means equal numbers.
Equal concentrations.
Concentration.
Yeah. Right. And so because we can run the simulation at any point, we have the data on how the concentration is spread out, and we can and this is just an illustration of different colors and different components. From that, we can identify how many phases coexist, in this case 2, and what the relative composition is. And there's some tricks to do that easily.
You'll be solving it in 2 dimensions?
I will be solving it in 2 dimensions because this allows us to systematically explore the parameter space. And I'll show you later that simple mean field arguments still match with 2D because we are not close to the critical point in these cases. Very doesn't matter. Yeah, and the numerical tricks involve doing some kind of spectral / Fourier tricks, which I'm happy to talk about offline. Good. So I've shown you a lot of things. So I thought maybe I'll stop and show you a couple of videos. So just to show how the simulations look. In this system, we have multiple components, 16 components, but the videos you're going to look at are only some random 2 of them. The scale bars represent their relative composition. So when they're really dark, their colors are either blue or green, that means they have dense phase. When they're light, they're dilute. I'm going to play a video which is going to show them phase separating. In this case, you see that the blue and green molecules form distinct phases. If you can see just by looking across here. And the dynamics exhibit a lot of the characteristic signatures of what you might expect in sort of more simple single component systems. And I'll just play that again to highlight a couple of features. You can see that there's Oswald ripening, small droplets dissolve to grow bigger ones. There's also coalescence and growth, which happens eventually between these 2 droplets. Yes, ma'am.
How many components is one of those white box?
I'm coming to that. So the question is, how many components are there in one of these? This is just showing you one field, but how many are there? And I will answer the question in the next slide. Thank you.
And the difference between the left and the right, these 2 realizations?
It's the same simulation, but we're looking at 2 distinct.
I'm sorry.
So this is species 1, species 2.
You said that.
Yeah.
Okay, and it looks like they're not occupying the same real estate.
Yes. So these are distinct phases. So you can follow this trajectory for a long time. And if you look at steady state and plot all their concentrations, it looks kind of tricky. So I'm just showing you this to show that there's a lot of information here. You can collapse it using the methods I've shown you before to see that there are 4 phases that coexist, which I have false colored here with these labels. And in each phase, and this is very hard to see, but I'm looking at how many components are enriched in a particular phase. This is an answer to Francis' question. And I just listed down the details here. So there's 4 in phase 1, 4 in phase 2, 5 in phase 2, and 4. So roughly about 4 to 5 components are enriched in each of the 4 phases.
And in all of them?
Yeah, I will solving. So the question is, are any of them in all of them? So the solvent phase, there's one solvent component that's non-interacting, that tends to be uniformly distributed. But the question of how these components partition into different phases is a lovely one. I'll answer that on the next slide.
I asked you before, I'm going to say more insight. Why are the diagonal patterns?
Yes. So first, the diagonal patterns are not universal. This is a particular trajectory where you see them. Second, I'm fairly confident these are because of the square box plus the periodic boundary condition.
What determines the angle?
Yeah, I don't have an answer for that.
Is it 45?
It always, so when they appear to be when they appear to be aligned, they tend to be either this way or this way along the diagonal. So it is reminiscent of a periodic boundary condition effect with the square box, but I haven't systematically dissected that.
If we competition into the third wavelength,
It does.
it's possible that you get an individual number of them along the diagonal, as opposed to some other direction. It might fit more naturally.
This is definitely possible. Also, if you wait for them to coarsen long enough, this will not be true. So eventual long-term coarsening, if they form only 2 essentially distinct phases, they will not be along the diagonal. But it's a good question. I have to dissect it a little bit. So this is just on a single trajectory, right? So the thing I wanted to do was ask how many of these features are general. The way we're going to do that is run multiple trajectories where each trajectory is randomly sampled from a distribution of chi. So the disorder is of French, if that's the right language, in the interactions. Repeat the simulations again and again, and then collapse the property. So what you see here in the solid black line is the number of phases over time. And you see that in this system, very characteristically across multiple replicate simulations with interaction parameters, that they all have a characteristic dynamics of how the phases evolve and grow over time. And there's a steady state value that's around 4 to 5.
What is the green?
Sorry, yes. The green line is just an example trajectory, one realization. Whereas the black line is the mean, and the gray shaded background is one standard deviation above.
And in all of these, you fix the signal diffraction.
Yes, everything is fixed in the simulation. They're only replicates because you randomly resample the interaction matrix, all the parameters of the vehicle.
Guess what would happen if you had a more asymmetric tie, so that most of the interactions were antagonistic or
Yeah.
the other way around.
So the question is, what would you guess would happen if chi was not a symmetric matrix?
Yeah.
And the answer is I wouldn't guess because I don't have any idea. It's a good question. I think in this system of molecular interactions, it's not obvious to me how non-reciprocity will emerge. In other active systems, for sure, that's true. I think one way to think about non-reciprocity is to go to the Dinner-Bay ensemble instead of Wigner's law. But because I'm not confident how even though there's a mapping to random matrix theory that I'll explain, I'm not sure how that will play out in dynamics.
I actually had a simpler question in mind, which is what would happen if your normal distribution was still a symmetric matrix.
I see. So if the distribution was skewed, yes. So my bet is that then all of these features are going to remain. And the reason for that, I will come to you soon. But as long as the distribution is, as long as the chi values are independent and identically drawn from a distribution, and the distribution has a finite variance, which is what is required for some of the properties of random matrix theory to hold, I suspect we will see the same result. No, okay. Okay, so this is the number of phases, but just on the previous slide, I had mentioned that each phase tends to be enriched in a typical number of components, and this is question of how do they partition between the different phases. So to systematically explore that, I try to measure how similar are any two coexisting phases. And the way to do this is to recognize that a phase, which for example is this yellow dot, is a point that's embedded in the dimensional space of concentration. It's uniquely characterized by its composition. So each phase, for example, this orange phase right here is enriched in 2 but depleted in 1 and 3. And this green phase is enriched in 1 and 3 but depleted in 2. So if I were to measure an angle between them, that would be basically 90 degrees, kind of orthogonal. And if phases look kind of similar, that there is a lot of sharing of components between phases, the angles would be close to 0. Because I have multiple trajectories and multiple coexisting phases, I can create a histogram. And what you will see is that it's an exponentially distributed histogram with its peak basically at 90 degrees, suggesting that most phases are orthogonal to each other. And the corollary of that is that each phase is roughly enriched in a typical number of components, here about 4 or 5, and those components are typically not shared across phases, again, consistent with the orthogonal. So
your finality test occurs at long times after everything is settled down. And when everything is, but when in your simulation, you never, what is the ultimate fate? One phase takes over, or then it would make sense to ask the question. So what final result? Some blobs floating around.
Yes.
So your box.
So what's the final point of a simulation of the question? And I think to the extent we assay steady state, which we can do by running slightly longer trials for lesser parameters, we see that there are a number of phases that predicted that coexist and they just keep coarsening. But as we all know, coarsening is power law, so you don't see very quick coarsening. But the number of phases seem to stabilize at long time. So there are multiple coexisting phases. And they have these properties, which I'll quickly summarize on the next slide, which is when they undergo spinodal decomposition, they form multiple phases at steady state, which typically have distinct compositions and are enriched in different kinds of components. I'll explain what I mean by state dynamics in a bit. You do see that the number of phases grows slowly. It's not like a sharp rate. And so the question we are wanting to explore is, can we try to begin to understand why this is happening? Like, can we explain sort of more principally why we see these features in our simulation?
You see 1/3 power law growth?
Yeah, we haven't systematically characterized, but the extent we have seen, it is roughly t to the power 1/3, especially at long times. The problem, of course, is when you have multiple phases, you have to really run very large simulations to get reliable power loss because they can really fluctuate from inference. So I'd say tentatively yes, but more work needs to be done to dissect. Okay, so one of the things we actually found in trying to understand why this was happening is we just systematically ran a number of simulations with different number of components, different shape of the distributions, sigma changes, different concentrations. And then we can, because we know the free energy of the system, we can compute what the initial instability looks like. We can compute the Hessian of the free energy and ask what are the different unstable modes. The Hessian is the second derivative matrix with respect to it's the matrix itself. So this is d squared f d phi I d phi j, and from its spectra, all the eigenvalues that are negative are unstable because they offer modes to lower the free energy. So we can count these, which I'm going to plot on the x-axis here in a bit. This is done at the beginning of the simulation, of the initial condition, right? And then on the y-axis, I'm going to plot the number of phases at steady state. And you can see here what's somewhat surprising, even though there's a big distribution around it, there is this rough law that tends to statistically hold, which is that each unstable mode gives rise to one phase, and there is a +1 correction here because there's overall mass balance, and there must be one of the phase doctrines for that. And so what seems to be true in our simulations, once again, statistically, not true at every simulation, but the realization is that every unstable mode tends to give you roughly one unique phase, and then one more for mass balance. And so the thing I can tell you is that, okay, let me show it on the next slide. So we want to understand why this is true. And so one of the things we can do.
Lower left there, there's a little where's zero?
Yeah, so the spectrum is a cartoon, but the straight line is 0, and the little peak is an entropic peak.
So that would be the maximum eigenvalue.
Yes, so the only thing that we can say, I mean, it's useful to know, and exact value is computable, but it's always going to be positive, so there's no worry about that because it's an entropic correction. And it scales with n squared. That's the number of components.
That's the positive value. So there's only one of them.
There's only one of them, and this actually comes from a correction to the Wigner spectrum that food loss derived late in the 90s. That shows that when you have because of the shape of the free energy, it's not true universally, because of the shape of the free energy we chose, there's only one single mode that will show up outside the spectrum.
So people with that portfolio would be very interested in that mode.
Yes. Yes, the question is not question. The statement is if these were stock portfolios, this mode would be useful to study. And exactly, yes. And so, that's the principle a lot of people have used when they use these Marcinko-Pasteur distributions as a null model to try to distinguish signal from noise, which again works well when the noise is again independent, unfolding. Okay, so this finding, which is that every unstable mode gives you a phase, is one we try to rationalize by saying that if you look at, because from Landau matrix theory, what we know is that the angle between any of the two eigenvectors of every unstable mode is going to be orthogonal, so they point in different directions. But I can tell you that if you do the algebra, the dynamics are not linear. They're non-linear, there's mass balance constant. So it's not obvious why this initial orthogonality still gives you distinct minima at the end of the free energy. And I've still not solved that problem. I don't know why that happens. The stable modes are also orthogonal, but they don't matter because, well, they don't matter for this dynamics at least because they're not going to exponentially grow. Yes, all modes are orthogonal in the because of the random matrix approximations. Yes, so we just use that as a way to try to argue why that might be a potential reason why each mode gives you a phase. But this would be exactly true if your dynamics are linear. But this is not the case here. It's non-linear, so there's still some mystery there.
Follow up on Grace's question: is your thought analogy preserved are the full nonlinear equations over time, or is it just initial analogy that's still no longer around?
So the reason why we believe it appears to be preserved is the following. is because the compositions at the end also seem to be fairly orthogonal to each other. You can ask a question whether the initial unstable modes point exactly in the direction of the final minima, and the answer to that is no. So the initial direction of an unstable mode doesn't tell you where the final concentration is going to be. But it is, on average, closer to the initial unstable mode than it is to other unstable.
Randomly drawn high-dimensional vectors always supposed to be.
If they're random in high dimensions, as long as you have randomly drawn vectors, they will be orthogonal. These are specifically the eigenvectors of a randomly drawn matrix, but that's the same principle. They are going to be orthogonal. Okay, so another prediction of this is that from the random matrix theory spectrum, what we know is that the eigenvalues tend to be equally spaced roughly. It's not that all of them have a characteristic size. And because of that, one of the predictions is that modes that have sort of more negative eigenvalues will demix quicker, and modes that have more milder eigenvalues will demix later. If you follow this principle that every unstable mode gives you a new. And you can actually, it's sort of more of a show and tell, but you can see this from simulations where this complicated axis is time, and this is drawn this way only because I am artistically challenged. The idea is that as you follow time, you see that initially that orange faces form, and then gray ones, and then pink ones, and then they eventually start coarsening. And this is just close to what we think is closer to steady state. So the number of faces has equilibrated. But you see that there's this characteristic time step, and it's again consistent with properties of the spectrum.
So again, you show that eventually this pulsation starts off between 1 plus the background and 2 plus the background. You can actually see it on.
Yes, and each one, another interesting note, because these are basically independent events. You can see that they don't demix from each other. And through analysis of sort of trajectories, you can find that almost all faces will demix only from the background, but just at different times. There's no correlation between everyone. Sometimes, I won't point that out because
So the composition that orange is the same too, is about the same one as the green or red.
yes, the compositions are fairly conserved. And in fact, the way I identify, I'm sorry, it remains about the same. It's only the sizes that differ. And of course, if you're very small, the composition is slightly different because of surface tension effects. But that's just a sort of known effect. I think we'll go to the next slide and say that because all of these statements have been true so far, to the extent we've explored, we thought we could just start predicting how many phases will exist at steady state by counting the modes. Exactly. We can do this because the Wigner spectrum is well known. And so we can write down an approximate equation for what that looks like. And so from the equation where the n is the number of species and sigma is the width of the distribution, it already predicts a couple of interesting things.
So, sorry, CEF is cumulative distribution function. Is that right?
Correct. So we can derive this formula by looking at
Semicircular.
the semicircular law.
Semicircular law.
Correct.
Centered where 0 is in the middle.
Yeah, so you have to find out basically how many eigenvalues can beat the entropic term, which is the correction. So it's not centered on, for the interaction matrix alone, the spectrum is centered on 0.
Okay.
But because there's an entropic correction that comes from the free energy, you're basically doing a calculation where you're asking how many eigenmodes are below the entropic correction. And that's the counting argument you will do. And again, because of the property of a random matrices eigenmodes, we know that the enthalpic term will always have half negative and half positive terms. So at most, only half of your modes can be unstable. Another way of stating it is that the maximum number of phases this formula predicts is order n over 2, and not more than that. So you can never, for example, reach the Gibbs space limit that we talked about in this system. And the part of the reason why I think this happens is because the Gibbs space limit 1 is a constraint. It's not a rule. It doesn't guarantee that it exists. But here you have lots of competing interactions because they're randomly drawn. There's frustration. There's no easy way for you to partition components into distinct phases because at least some of the other components will be attracted to them. I think that's possibly a slightly wishy-washy, but kind of my favorite explanation for why this happens.
So finally, about this phase will be that then the max equals n.
Yes.
That would be what.
And there is one guaranteed way, at least in this system, to get that limit, which is basically make every component completely dislike every other component with a high enough energy scale, and that's what we should get. But I think here what we have is, because the competing interactions is a reduction in this. Good. The second point I want to note is that if you have a fixed distribution, as you keep sampling more components, you're not going to get more number of cases. And the reason is that, well, mathematically, you can see that there is a positive term outside, but a negative correction that scales with n inside. So that gives you this trade-off. The intuitive reason is that as you keep increasing number of components, you start diluting the other one. And so that's the drive against demixing. And at low n, the number of phases is limited by the fact that you have this n+1/2 limit. There's usually a peak somewhere. And because we have done simulations across very many conditions, that y-axis is number of phases at steady state, next is number of components, and each of these curves represent different distributions with different widths. The straight lines are theory, and the dots, triangles and dashes are simulations. You see they largely agree, except when sigma is kind of low. And the reason for this, and I'm not going to show you the data, is that there's finite size corrections that happen to the spectra. So our prediction of what the spectra looks like from Wigner's distribution is not accurate. And so we can actually start to make some corrections based on Tracy-Widom extreme statistics, and that helps us at least bridge this gap. Part of the reason is finite size effects. Okay, but this tells us basically that if you keep having more components from a fixed distribution, there's a maximum amount of cases you can enter. If you wanted to do more than that, the distribution has to start widening. The sigma needs to scale with n, and at least with square root of n, to get you more than linear encoding of number of phases. Again, the data shows simulation and theory largely agree. So I have some ideas on how you might design ensembles with widening interactions, but I won't discuss them today.
So another problem is where making sure you have a box distribution, electrical box distribution. The results don't change for large n. Is that expected here?
I don't know what specifically the box distribution is.
So it's sort of it's literally a box.
Oh, like a square distribution.
A square without a
Yes.
maximum.
Yeah. So theoretically, what I can tell you, the spectra is, and seen this without doing the experiment of the simulation proof, is that as long as the spectra is not long-tailed, you're going to get these. As long as the chi's are independent and identically drawn, all of these results are going to hold true. The only complication is that, the only complication is when you have long tails, because you can't guarantee the variance of the distribution. Yes. All right. Good. So I'm getting close to the end of time. I'm saying, so let me actually introduce one slide, which I added because I remembered that this was an active matter seminar, and everything so far is not active.
You got to go over, say it's part time.
Oh, it's 20 minutes.
Yeah, like 15, 20 minutes.
Oh, okay. So I have some time. Okay. Okay, good. So token activity slide. There's more active stuff in the second part, but because this system was interesting, we thought we'd explore what happens when you add activity, more specifically chemical activities, identical to everything I've said now, except every component is being actively produced and degraded. And the simple sort of approximation they're going to make is that there's only one rate determining number for all the components. They're all produced and degraded at some typical reaction rate, which is K. And once you do this, you can go through all the stuff I've done before. And what we find in general by looking at the spectra again is that higher activity, by which I mean faster cycling, is going to suppress phase separation. The number of phases will go down as you increase the activity. And it goes down in some, with some linear approximations, it goes down linearly, but this is not exact. Answer your question.
Well, if you go then to explain why you don't reach the distribution, because you're not really going to do it.
Well, the question is, this is explained.
You have fewer things, right? Because this is active and
Yeah, so the question is, do you have fewer phases in the Gibbs limit because of this? So the Gibbs limit argument holds true irrespective of whether you have activity. So the limit on n plus n over 2, that happens even in systems with zero activity. Purely passive systems. But here, what we find is that as you start cycling components, you're going to reduce the number of phases. The x-axis is a measure of activity, the y-axis is the number of phases, and you can tunably go from 6 to just a completely demixed uniform phase as you tune the rate of cycling. And that's simply because every time a component is being produced and degraded, it loses spatial correlation. It no longer knows it's sitting in a phase. So it counteracts diffusion. It acts like diffusion. There's a bunch of theory to this. Anyway, so I'll just quickly summarize by saying that this part of the talk, we started by reformulating and asking, when you have complex mixtures, but you make an approximation that the components are randomly interacting. Can you say anything about their statistical mechanics of the phase behavior? And what we find is even in such what might call sort of simple null-like models, you can get very rich phenomenology with multiple coexisting phases, very characteristic dynamics with distinct compositions, and a random matrix theory starts to have make connections between what we observe in simulation and sort of the mechanistic theory.
I don't want to hold you up if you don't have a stick, but can you say anything about the interface?
Yeah, so the question is, can you say something interesting about the interfaces?
I'm sure.
Yeah, so the sharpness of the interface is not particularly interesting from what you've seen because I can tune that basically by changing the relative surface tension. And I'm working in a limit where I assume surface tension is kind of important because it helps with numerical stability because it's a fourth order correction term at long wave numbers, wave lengths, wave numbers. But yeah, so we're not systematically enforced. We are working generally in the limit where interfaces are sharp enough that it matters to get you stability at long. And we don't see any large corrections when we change. That happens.
So,
yes. Correct. So there's a what's the effect on composition? So there's basically a Laplace correction to the composition. We haven't basically it's hard to estimate these things because we actually don't know the steady state, the true steady states. All of them have been simulated to the endpoint. So it'll be interesting to go back purely from the free energy, check where the minima are, and ask what the changes are. That's something we are working on now. It's a little bit tricky because of the dimensionality involved.
And to follow up on that, if I remember correctly, in the original model, you have identical coefficients of the gradient of the composition squared for all components. So that's part of the piece.
It's absolutely.
You could take those to be random too.
Yeah, so the question is, I assumed everywhere that the surface tension is equal and that could in potential be random too. One argument for why they don't change too much is that in vivo at least, even though this is widely far away from biology, one of the major determinants of surface tension appears to just be kT over the length of the molecule squared. So that seems to be a fairly good determinant of surface tension. So it's just the edge, literally, the size of the molecule that it can.
However, you can have molecules with vastly different sizes.
Indeed, you can have molecules with vastly different sizes. It turns out that it depends sensitively on your definition of vastly different. It turns out so most things that have been known to now are roughly between 5 to 10 nanometers. So, but it's certainly an important point. And there's lots of places that you could add quench disorder, right? Compositions can vary, other stuff that can vary too. So I will, because I have very little time, maybe I'll quickly run through the second part of the talk, which also is a paper we just wrote earlier this year. And this is the opposite direction. So instead of starting bottom up from a model, we're going to be inspired by observations made in cells and in systems in biology and ask whether using principles of phase separation and activity is going to teach us something.
So this is great. Can you just summarize briefly when you said it, and this is what is the usual method for when you introduce activity?
Yes. So the take-home message from the take-home message for when you have components that are active in the sense that they're being degraded and formed is that they basically always reduce the number of phases that coexist at steady state, and that there is a theoretical connection that you can make that is length dependent. It depends on the size of the box, that allows you to figure out how much activity will translate to how much of the unstable modes you find. Basically, the idea is that all the remaining modes that you have not quenched are still unstable and orthogonal to each other, so they will give you. That's the paper.
And also, like other problems, small number of components, the rocket stop growing.
The size, yeah, the good point.
Size stuff.
There's another point I have not mentioned here is that you get arrested ripening as well, or arrested coarsening.
Yeah.
And this has also been explored in the context of two-phase systems, but we see the same phenomenology in multiple coexistence. Yeah. All right, so someone has got to keep me on time, so I'm going to go ahead and talk about this in some, at least begin to introduce what the important questions are, and whether we answer them or not is secondary. This work was done with Arup and many others who are this very talented team of our experimental collaborators who did a lot of chemistry and work in single cells and using very complicated imaging techniques. Okay, so I thought I'd give a quick primer on why we are focusing on the nucleus, and it's sort of introducing a puzzle. It's a classic puzzle which we've known for a while, which is because of we all have a common genome, right? All humans have a common genome, a shared genetic blueprint. But despite this shared genetic blueprint, we are able to make lots of different cell types. The way we do this is not by having lots of components. In fact, only 1 to 2% of our DNA contains code for genes or components. We actually have very extensive blueprints. We have about 10% of our DNA that's non-coding. It basically provides instructions to zeroth approximation. Many apologies to the developmental biologists in the audience, which allows us to mix and match these components in different ways, much like a Lego set, to build very different cell types. So understanding how gene expression is regulated, which is sort of figuring out which paths are used, is very important to figuring out, it's essential to understanding cell identity and physiology. And so one of the things we have started to learn, and it's been known for a while now, is that many processes in the nucleus, which is where the genome is, are compartmentalized in these kinds of liquid droplets. And so here's an example of 3 different droplets. One is transcriptional, one is called the nucleus speckle, and the other is the nucleolus, which I introduced before. So the green dots are particular proteins that are enriched in these droplets. I'll show you an example of a video. These are really dynamic and small assemblies. You can see some of them peeking in and out of existence, others that are just rapidly moving around, all within the time scale of a minute. And the scale bar is missing here, but all of these are either diffraction limited or about half a micron. This is a maximum intensity projection. This is not going out of plane. So when you see something blink out, it blinks out. Yes. This is a maximum intensity projection done at 3x of a mouse embryonic stem cell. And the green color blobs are of a mediator, which is an important molecule in transcription activity. So what's common to all of these assemblies is the following, is that they're all sites of transcription where RNA is produced. And so there are 2 questions that are conceptually interesting, right? Which is, how does RNA, in fact, generally modulate the phase behavior of these protein assemblies? More specific question, how does the fact that you have active RNA synthesis tie into the properties of the condensates and maybe tie into the properties of the function? And so we were interested in exploring this because there's a couple of biological puzzles that have not been answered for a while. And that is related to the following point. I told you earlier that genes, which make the paths, the proteins, only make a small percentage of a genome. And when they're transcribed, they're converted into messenger RNA or RNA. What's been known in the last decade is that this is not the only place at which transcription happens. There's lots of other elements, regulatory elements, that like the promoters or the enhancers, that also have low levels of short RNA, widely known as non-coding RNA, enhancer RNA, antisense RNA, that are quickly produced, often degraded fast, and their role exactly has not been well understood. And it is known that if you try to suppress these, there's an effect on gene activation. So while there are some correlative effects, what's the mechanism by which transcription of all these non-coding DNA elements affect gene expression is not well understood.
I think on the way, 2 strands of DNA that are actually connected off diagram?
Yeah, so I should, yeah, so let me, the gray parts of the chromatin of the DNA background, all I'm pointing out with this is to show that the enhancers can typically be very far away in linear sequence space, but they often come back closer in 3D space. So we are eventually connected.
Move around and maybe bind the promoter or?
Yeah, so that itself is a complicated question, whether they actually physically bind to the promoter or not. In fact, most of our data in recent time with super resolution seem to suggest that this distance is ordered 10 to 100 times molecular sizes. So they're probably not in physical contact. There's a world of questions here. I'm sort of leaping over all of them, but there's an interesting question too, which is, this is where a lot of the droplets are forming, like spectrals or transcriptional condensates. We can show by fish that these things are close to particular genetic loci. But there's a question here that becomes a little confusing. If you have hundreds of molecules that promote transcription in here, how do you make the gene stop expressing itself? And the reason why this is an interesting question is, phenomenologically, we know this happens. That is, gene expression happens in bursts. And there are lots of simple and classic models, for example, the telegraph model by Uri Alon, which explains why this happens if the rate limiting steps were just single binding event that allows you to turn on or off the gene. But when you have hundreds of molecules that can promote transcription, how exactly you can still get this discrete bursty expression of genes is not very well understood. So these are the questions which I will be trying to answer through the lens of exploring how RNA activity might modulate phase behavior.
My question is, we have around like 10 or 15 minutes.
I have about 10 minutes.
Yeah.
Okay, good. All right, that's good, because I think I can make some headway. Probably even finish.
Don't have to talk fast.
So, the first question is to ask, what is the interaction between RNA and protein, and how does that translate to phase behavior in general? And this is a topic that's actually been studied for nearly 100 years. There's lots of references. Initial proponents were Haldane and O'Pirin in the 40s and 50s, where they were very fond of a hypothesis of sort of origin of life models of how RNA world was existing, how RNA and amino acids can form complex coacervates. So I'll tell you what they mean.
What is it called?
Yes, I will get to that. Right. So a complex coacervate is specifically a droplet that forms between species that have different charges, and the interactions are mediated by the electrostatic interaction. And so a phenotype or a consequence of complex coacervates, but more generally when there are interactions across molecules that make them stick to each other, is that if you have too much of one molecule or the other, they're going to suppress phase separation. But if you have relative stoichiometries where you can promote interactions, that's going to promote association with the droplet. With RNA and electrostatics, it's easier to see, right? You have the right stoichiometries, you have enough attractive interactions that dominate, but if you have too much of one, they cannot compensate the repulsion. So this was the model we thought made most sense. And my lovely collaborators also tested this out first in the test tube. So they could purify a part of a transcriptional protein, here mediator, and then titrate different concentrations of RNA. And then you can see that droplets start forming after some threshold. And as you add more RNA, they end up dissolving again. And if you were to measure phase separation, which I'm just showing you here as partitioning, and then just simply plot in blue what the ratio of the charges are between the two components, because you know the sequence and you know how much concentration you have, you see that phase separation is maximally promoted when their charges are equal stoichiometrically balanced. So it's consistent with the idea that electrostatics is the dominant driving force. But this is still in a test tube, right? So the cell, what we know happens is these droplets form, and there are likely sites where there's activity that happens. So question we wanted to ask is, can we ask how active RNA synthesis impacts phase behavior? And as people who might have worked on coacervates might know, writing down thermodynamic descriptions faithfully are quite hard because of the nature of the interactions. So we, instead of going to write an exact model, we just took a Landau approach with the two order parameters being phi p and phi r. They are proxies for the relative concentrations of total protein and total RNA. And we write down a free energy that's, perhaps I will not go into this in detail. And the only point I'd like to mention is you need to have certain fourth order terms included that allow you to create this re-entrant phase behavior. And so that's just what happens in the absence of any activity. That's just how you write down the equilibrium properties of RNA and protein. But you have to account for the fact that the dynamics that produces them, dynamics of diffusion and degradation. And so you can write down coupled dynamics that tracks how the protein and RNA evolve. And I guess the one important point to note down here is that protein is conserved, and this is because protein degradation is fairly slow. So on the timescales we are interested, it's okay to write down a conserved model. It's definitely not true for RNA. You can write down these equations, you can non-dimensionalize them, and you can find that basically some equivalent of a Dam-Kohler number, it measures what's the rate of activity over diffusion, is important for figuring out how much basically the fact that you have activity matters versus you just simply mix these components in a test tube at equilibrium. And because we know some of these numbers for transcriptional systems, we can estimate them to an order of magnitude. You can see the range is quite large, but the fact that it's quite high seems to suggest that reactive terms are very important and rate limited in vivo, which means local concentrations are very important, not sort of global problems. Sorry, yes, so F is the free energy. So I'm just writing down the diffusive chemical potential of the protein. dF will be 5. Sorry, it's a good question. Okay, so I'll quickly summarize the main finding from the study, which is one of the things that is known from the biology is that early stages of transcription, known as initiation, is characterized by low rates of transcription. So the rate of production is low, and there's lots of non-coding RNA. And if you run simulations, you would see that with low rates of RNA, that leads to condensates that glow with local concentrations that are diffused and stuck near the condensate. And if you have to increase the rates, as is known for productive elongation, what happens is that you get a sudden burst of RNA that gets concentrated, but because it's all locally concentrated, because of the ratio of reaction to diffusion, that starts a feedback loop where it starts melting the droplet. So this is our model for how you might potentially have sort of cyclic starting and ending of transcriptional condensates, where there's a transcriptional burst that starts because there's low amounts of RNA. And as you start producing more and more RNA, you can start dissolving it because you have very high local concentration. And that gives you a cycle of typical numbers of RNA that's produced. And a prediction of this model is that if you had a drug that could somehow decrease fast rates of transcription or elongation, then you would move this way along the axis, and all your droplets in the cell should grow bigger and become more stable. And this is exactly what we see. By we, I mean my lovely collaborators perform experiments where they added 2 different drugs, both known to suppress productive elongation. And in the green, again, are these transcriptional condensates, which you can track over multiple cells. And statistically, you see that their size goes up across the entire distribution by a factor of 2 or 3. For many of the other droplets, which I earlier pointed out were flitting in and out, they were stable only for a few seconds or a minute. There are parallel experiments we did with PCPalm, which is a temporal super resolution image, and it allows us to sample how those properties change as well. And that too, we saw stables became bigger and more stable, faster. Okay, the final experiment I'll mention is one that's focused at a specific loci, because all of the data I've shown so far tells you what happens across the entire cell. So there are confounding effects that may potentially carry on. So here, my collaborators set up a lovely experiment where you could put out a gene that was located close to an RNA and stably integrated into the genome. And then you could tunably increase how much RNA is produced close by. So as you change this RNA concentration, you can track out how much gene expression is happening here. So if local RNA concentrations are important, there are predictions from the simulations that can give us idea of qualitative trends, which is that if you have very short RNA, can't ever build up high concentration, so you never suppress gene activity. But as you increase the length of the RNA, you go from being promoting gene activation to being suppressed. And it's consistent with this sort of complex coacervation subject to local activity model. And I'll quickly say that it's the same trend we observed in vivo too, in cells, where if you track the gene expression on the y-axis, and the x-axis is a proxy for how much RNA transcription is happening, we see that as you increase the length of the RNA, which in turn changes the local charge concentration, the more you have, it becomes more repressive instead of being promoted. But there is this distinct trend where initially it activates genes and then suppresses them later. I think I will stop by saying that by getting inspired by the biology, but also building a model of complex coacervates, a model of phase separation, and then incorporating activity, I think this allows us to start giving a possible role for why there is non-coding RNA that's initially present, and how you might still get discrete bursts of transcription even when you have lots of material present locally. And there are some problems that have historically been sort of given correlated data for, which is genes that are in nearby neighborhoods seem to be regulated together often, even though there's no biochemical interplay between them. And so this transcription might be a potential way how this co-regulation happens. A part I didn't talk about is the same kinds of activity, if they were localized in space to a small extent, you get a number of non-equilibrium morphologies like vacuoles, you get deformed droplets that form due to the fluxes, you get droplets that move. And a lot of these are interesting from sort of active matter perspective, but also for over 20 to 50 years, many of these morphologies have been documented by cytologists and embryologists looking at cells, but they've never been explained with a unifying model. And so this is potentially a model that gives us insights into how the cell nucleus is organized. And with that, I think I will stop.
Thank you. So, yeah.
I was wondering if you want to cellular special
With the what, I'm sorry? Vishal ensemble, yes. Okay.
So that was
Right.
And I'll probably have to keep these little phases. And so I was wondering if you guys are planning to look at correlated institutes, how you would introduce correlation, and what do you expect? How do you expect that would be?
Yeah, it's a lovely question. So I'm just going to summarize the question broadly as what happens when you introduce correlated random matrix ensembles, for example, the Vishal ensemble. And it's one that we are deeply interested in studying for the following reason. When you want to make libraries of molecules who interact, when you sample them, they are not independent, right? When you sample them, there's correlations between them. So studying how correlated molecules systems give you resultant phase behavior is very much of interest. The issue, of course, with correlated variables is you can no longer guarantee orthogonality in the eigenvectors. So we are, we can study this from a computational perspective, and that's what we're starting to do, but we haven't really sort of having any theoretical insights on that.
So that's my thought. So thanks for the answer. As a follow-up to that, so for example, for the Wishart matrix, you expect the elements are correlated, it's still real symmetric matrix. So the eigenvectors would presumably still be orthogonal.
I don't. Okay, so do all real symmetric matrices have orthogonal eigenvectors? Yes. That's a good point. Okay, I haven't thought about it too deeply. One thing we have started to explore is what happens when you have explicit correlations introduced by making designed interactions. And in those limits, what we find is that there's sort of basically a trade-off between the strength of the design interaction versus the strength of the noise. But I haven't explored the Vishal example. That's a good point.
Can you hear me, Krishna?
Yes, I think so. Yes.
Okay, thanks. That was really interesting. I have two questions. One is someone asked earlier when you were setting up the premise about one might argue that biological molecules are finely tuned such that this random distribution is not representative of their interactions. But it seems like you could flip it and ask, if you do, like, I'm sure there are in vitro experiments, right, where you put lots of components into a dish. Does your model correctly predict or not the number of phases that you get? I.e., can you test whether or not biological molecules that do phase separate really are just kind of a set of random interactions?
Yeah, it's a good question, right? So, everyone hear the question? Yeah. So, from an experimental perspective, you still haven't gone there, surprisingly, because while we started, so most of the in vitro interactions model have focused on a handful of proteins. And so we don't necessarily have a great model of trying to ask, if I have a sort of wide distribution of molecules, can I robustly classify how their interactions are? And more specifically, often these interaction studies study binding. And in phase separation, ability to interact is correlated with binding, but what you really need is something closer to a virial coefficient. And this has not been always easy to study. But that being said, that's sort of more of an excuse than a reason. So there are models, statistical polymer physics models that allows you to ask, when you have two types of sequence, what's a rough virial coefficient you get? So you could ask this question based on some biologically motivated sequences. Can I design an ensemble and ask what its properties are? And these are experimentally realizable. These are very much on a to-do plate, but have not been done.
Cool. And then my second question is, in terms of the point you made toward the end of the first section on activity and how that alters the number of phases, has it been noted that, I'm not so much thinking about like the nucleolus, but in like cytoplasmic phase separated organelle type things, are the components of those, have they been noted to be faster degraded or more slowly degraded? Any kind of like unique aspect there?
So if we focus on the cytoplasm, I'm not sure I can give you a complete answer, because some of the most prominent cytoplasmic granules are the ones that form in response to stress. And those usually serve as storage organelles. Almost translation, transition is all sort of hindered that. So there's very low activity there. The nucleus, of course, is a hotbed of activity. And there are examples. For example, proteosomal clusters form in the nucleus, and they are sites where there's rapid protein degradation. Whether this is universal or specific to a certain set of molecules has not been studied. And I think the best example of how activity regulates condensate properties still remain synthetic. There are a number of physical chemistry labs that have synthetic analogs where they can modulate activity. And they show a lot of rich behavior, even with a couple of components. But all of it's done with non-biological molecules in vitro.
Thanks.
Thank you for the question. Okay, looks like we're out of questions. Thank you, speaker, once again.
Yeah. Thank you.